Imagine turning your own body cells into a precision medicine, um, a microscopic army trained and supercharged to hunt down and destroy cancer. Picture your immune system, our most potent defense, not just reacting, but proactively, like intelligently seeking and destroying a specific threat. That's the revolutionary concept behind SCORE T cell therapy, and it's what we're diving into today. Welcome to the deep dive. This is where we take a stack of your sources' articles, research our own notes, and really try to extract the most important nuggets of knowledge or insight. Our mission today is to give you a shortcut, to being truly well informed about one of the most exciting and frankly transformative advancements in modern medicine, CR T cell therapy.
Indeed, it's a truly remarkable and transformative approach in oncology. It really represents a paradigm shift. We're leveraging the body's inherent defense mechanisms, but in a highly targeted and incredibly powerful way. In this deep dive, we'll journey through its fascinating origins, unravel the intricate science of how it works, understand its current profound impact on patients, and peer into what the future holds for this cutting-edge treatment.
Okay, let's unpack this. It's definitely a complex topic, but one that offers, well, immense hope. From the initial breakthroughs in the lab to its incredible journey into the clinic, we'll follow the story of these remarkable cells. We'll try to unravel both the scientific ingenuity and, the historical context that brought us to this incredible point in medical innovation. So for those of us who might be new to this, let's start right at the beginning. What exactly are T cells, and what fundamental role do they typically play in our body's immune system?
Right, T cells are absolutely vital components of your immune system. They're a specific type of white blood cell. You can think of them as the uh, elite specialized forces of your body's defense system, almost like a sophisticated surveillance team.
Hmm.
They're constantly patrolling, monitoring your body for any signs of abnormality, whether that's an invading virus, bacteria, or, yes, rogue cancer cells. And they achieve this critical function by recognizing specific protein markers, we call these antigens, that are displayed on the surface of these abnormal cells. They do this using their own unique surface proteins or receptors.
Okay.
Now, once a T cell's receptor successfully identifies an abnormal cell, that T cell becomes active. It gets switched on, so to speak. It then springs into action, directly destroying the identified threat, and importantly, simultaneously signaling for other immune cells to join the fight, creating this powerful cascading defense.
So, if T cells are already these incredible immune soldiers, here I are T cell therapy sounds like we're giving them a significant upgrade, almost like a superpower. How exactly does that work? Are we enhancing their natural abilities or giving them entirely new ones?
Precisely. Yeah, you've hit on the core idea. It's a significant upgrade. A superpower is a good way to put it. Chimeric antigen receptor, or CRR, T cell therapy is a highly advanced type of immunotherapy. Essentially, a new, specially designed lab-made gene is introduced directly into a patient's own T cells.
Okay, a new gene.
Yes. And this genetic modification isn't just a minor tweak. It's a profound re-engineering. It fundamentally changes their targeting ability. This new gene equips the T cells with a novel receptor, the chimeric antigen receptor. And this engineered receptor is specifically designed to enable the modified T cells to more efficiently and precisely detect and crucially kill cancerous cells.
Right.
Cells that their natural receptors might not recognize as effectively.
Right.
It gives them this laser-focused ability to find and eliminate specific enemies that might otherwise evade detection.
That sounds like a fascinating journey for the cells themselves, but also a very involved process for the patient. This isn't just a simple pill or a quick injection, is it?
No, not at all.
Can you walk us through the practical steps? What might a patient experience from start to finish?
It is indeed a multi-step, intricate process. And yes, it truly is a significant journey for the patient. The first step involves isolating the patient's own T cells from their blood. This is what we call an autologous treatment, using the patient's own cells. In some specific rarer cases, T cells might be sourced from a healthy donor, which is termed allogeneic treatment, but mostly it's autologous.
Okay.
This isolation process is called leukapheresis. During leukapheresis, a specialized machine, kind of like one used for blood donation, draws some of the patient's blood. It carefully separates out the white blood cells, including the T cells we need, and then returns the remaining blood components back to the patient's body. This is typically done via a central venous catheter, which is a temporary line inserted into a large vein just to make the process more efficient. So once these white blood cells are collected, they are rapidly transported to a highly specialized cell processing center. And these aren't just any labs. They are state-of-the-art facilities designed for incredibly precise cellular manipulation and engineering under sterile conditions. Very high tech.
Right.
Here, the collected T cells are stimulated and expanded into incredibly large numbers, millions, even billions of cells. To drive this significant expansion, they're typically treated with specific agents, like the cytokine interleukin-2, IL-2, and anti-CD3 antibodies. These act as signals to encourage their growth and activation.
Hmm.
This entire intricate manufacturing process, from the initial blood collection to having enough modified cells ready for reinfusion, can take a significant amount of time, usually anywhere between, say, 3 to 6 weeks.
3 to 6 weeks, that's quite a wait.
It is. And this waiting period can be a very challenging time for patients and their families, naturally.
I can imagine.
So, during this crucial waiting period, while their specialized T cells are being engineered in the lab, patients might receive what's called bridging therapy.
Bridging therapy.
Yes, this could involve traditional chemotherapy or radiation therapy. It's strategically administered to help manage their existing cancer and prevent it from progressing too rapidly while they wait for their personalized cellular treatment.
Okay, makes sense.
Then, crucially, just before receiving their modified CAR T cells, patients undergo another round of chemotherapy. But this time, it's known as lymphodepleting chemotherapy.
Lymphodepleting, so reducing lymphocytes.
Exactly. This isn't primarily to fight the cancer directly at this point, but rather to prepare their body, almost like clearing the field. This pre-treatment suppresses their existing immune cells, making space for the new, powerful CAR T cells to engraft and thrive. And it also helps prevent their body from rejecting these potent new cells.
Okay, so that's a critical step before the actual CAR T cells go in.
Very critical. Then comes the final, truly momentous step. The actual infusion of the new CAR T cells back into the patient. This is done via an intravenous line, much like getting a standard blood transfusion. The infusion itself is relatively quick, typically taking only between 5 and 30 minutes.
Oh, that's faster than I expected.
The infusion itself, yes. But, and this is important, due to the potential for serious, immediate side effects, patients often need to stay in the hospital for about 7 to 10 days immediately following the infusion. This is for close, intensive monitoring by specialized teams.
Right, makes sense given how powerful these cells are.
Absolutely. And even after discharge, they're advised to remain within driving distance of their treatment center for the first month. Plus, they require a 24-hour caregiver to be with them during that initial month in case of any delayed side effects.
Wow.
And often, they need someone else to handle driving for them for up to 2 months after the treatment. It really underscores the intensity of this therapy and the absolute importance of robust post-treatment support.
That's truly remarkable, taking your own cells and transforming them into a highly targeted living medicine. Given its complexity, what specific conditions has this therapy been approved to treat so far? And what does the number of approvals tell us about its impact?
Right now, CAR T-cell therapy is primarily an option for certain aggressive types of blood cancers, often when other standard treatments haven't been effective, or if the cancer is unfortunately returned after initial remission. It's really a breakthrough for these specific patient groups.
Okay, so mainly blood cancers for now.
Yes, the list of blood cancers it's approved for includes B-cell acute lymphoblastic leukemia, or ALL, especially in younger patients, also diffuse large B-cell lymphoma, follicular lymphoma, high-grade B-cell lymphoma, mantle cell lymphoma, multiple myeloma, and primary mediastinal large B-cell lymphoma. These are all cancers of the blood or lymphatic system, which are generally more accessible to these circulating immune cells compared to, say, solid tumors. And the rapid progression of CAR T-cell therapy is perhaps best seen in the number of approvals. There are 6 distinct therapies now FDA approved in the U.S. For all L in children and young adults, there's Tyrogen Lekuso, known commercially as Kymriah. Brexicabtagene autoleucel, or Tecartus, is approved for mantle cell lymphoma. Axicabtagene ciloleucel, known as Yescarta, is used for several types of B-cell lymphomas.
Okay.
Then there's lisocabtagene maraleucel, or Breyanzi, also for diffuse large B-cell lymphoma. And finally, for multiple myeloma, we have itacabtagene vicluso, or Bacema, and ciltacabtagene autoleucel, marketed as Carvykti.
6 different therapies already. That's significant progress.
It really is. Each of these approvals represents a major milestone, offering new hope, and for some patients, even a potential cure for cancers that were previously very difficult, if not impossible, to treat effectively. It truly underscores the incredible precision and development happening in this specialized field.
Okay, this is where the engineering truly gets fascinating for me. Once scientists have isolated these T cells, how do they actually go about building or engineering this chimeric antigen receptor? What are the key components of a CAR T cell that make it so effective at its job? It sounds like molecular architecture.
It really is. The CAR itself is a marvel of bioengineering. like a sophisticated custom-built antenna on the T-cell surface with a built-in activation switch. It's not just a single component, but rather an engineered receptor designed to combine two key parts. First, an antibody-like external domain. That's the part that specifically recognizes the cancer antigen on the tumor cell.
The targeting part.
Exactly. And second, an internal T-cell signaling domain. That's the part inside the cell that tells the T-cell to activate and kill once a target is found. But there's a crucial structural component that often goes unnoticed, but is absolutely essential for the CAR's function, the transmembrane domain.
Transmembrane. Crossing the membrane.
Precisely. You can think of this like the foundation of a building or a robust anchor. It's a part of the protein, usually a hydrophobic alpha helix, that spans the T-cell's outer membrane. It securely bridges the extracellular antigen recognition part, that antenna, searching for cancer cells, with the intracellular signaling region inside the cell. Its role is fundamental. It ensures the overall stability and proper function of the entire receptor, making sure the T-cell can effectively receive the signal to attack when it binds the target. Some designs, like those using the CD28 transmembrane domain, are known to result in a receptor that's highly expressed and very stable.
So it's clear there's a lot of science and meticulous engineering that goes into ensuring the CRR is stable and functional. But is this a one-and-done design, or are scientists continuously innovating? Are there different generations or approaches to designing these cars?
Oh, absolutely not a one-size-fits-all solution. The field is incredibly dynamic and rapidly evolving. Scientists are constantly refining these designs to make them safer, more effective, and more versatile. One notable advance is the Supra car. Supra stands for split, universal, and programmable.
Split, universal, and programmable. Okay, that sounds flexible.
It is. This is a fascinating leap because, unlike earlier cars built to target just one specific antigen, SupraCar allows for much finer regulation of its activity. This helps to limit potential overactivation, which can cause side effects, making it potentially safer and more controllable. And what's truly revolutionary here is that it can be programmed on the fly, essentially, to target multiple different antigens without requiring further genetic modification of a person's immune cells after the initial engineering.
Wow, how does that work?
Well, the CureR itself is split into two components that have to come together to activate the T-cell. One part is on the T-cell, and the other is a separate adapter molecule that binds to the cancer antigen. You can give different adapters to target different antigens. This flexibility is particularly useful for treating tumors that are antigenically heterogeneous, meaning they have a mix of different antigens on their surface. You can administer a mixture of desired antigen-specific adapters, effectively creating a multi-pronged attack.
That's incredibly clever. Targeting tumor diversity?
Exactly. And beyond engineering the QR itself to be more flexible, scientists are also thinking about how to control its activity even more precisely, ensuring the T cells only activate exactly where and when they are needed, minimizing collateral damage, so to speak.
Right, the off-target effects.
Precisely. And that's where innovative approaches involving bispecific molecules as switches come into play. These are ingeniously designed molecules engineered to target two things simultaneously. One, a tumor-associated antigen on the cancer cell, and two, importantly, a specific molecule on the surface of the T cell, like CD3.
So it needs both connections to flip the switch.
Essentially, yes. This dual targeting acts like a crucial safety switch. It ensures that the T cells only become fully activated when they are in incredibly close physical proximity to a tumor cell. This minimizes off-target effect on healthy tissue that might express the tumor antigen at low levels or accidentally activating T cells elsewhere. For example, an anti-CD20-CD3 bispecific molecule has shown high specificity to malignant B cells in mouse models. Similarly, another molecule called FITC can effectively redirect and regulate the activity of FITC-specific SAR T cells towards tumor cells that express folate receptors. It shows the incredible precision possible with these molecular switches.
Amazing control.
And the innovations don't stop there. Recent advancements have also focused heavily on incorporating alternative activating domains within the CRR construct itself.
Inside the cell, you mean? The signaling part.
Yes, the part inside the T cell that actually transmits the go signal. Think of these as different types of internal booster signals or fine-tuning mechanisms. The goal here is to enhance the CAR T cell's efficacy and help them overcome resistance mechanisms, especially in those notoriously challenging solid tumors we mentioned. Researchers are exploring various internal signaling components, for instance, incorporating parts of Toll-like receptor 4, TLR4, signaling, can significantly boost T cell activation, improve their proliferation, and enhance their persistence within the body. Similarly, using the FYN kinase can improve the signaling cascade. Even using parts of CAR receptors, like CAR2DS2 with a DAP12 adapter, has shown improved T-cell proliferation and anti-tumor activity in studies. They're even looking at non-conventional costimulatory molecules like MyD8-ATD40.
So tuning the engine, essentially.
Exactly. Creating smarter, more resilient CAR T-cells, capable of fighting tougher, longer battles against cancer.
That is truly sophisticated engineering. It sounds like the manufacturing process itself is also undergoing major advancements, or maybe a glow-up, as the outline suggested, aiming to make this therapy more accessible, perhaps even faster, and maybe more affordable.
Yes, exactly. That's a huge focus. Due to the inherent complexity and, frankly, the resulting high costs of CAR T-cell therapy, there's significant effort across the scientific community to drastically improve manufacturing processes and reduce these burdens. One really promising strategy involves in vivo CAR T-cell manufacturing.
In vivo, so inside the body.
Correct. Modifying T-cells directly inside the body. This could potentially eliminate the need for that complex, expensive, time-consuming external processing in specialized labs. That's a huge logistical and cost hurdle.
How would that even work?
Well, a key aspect is in situ modification. T-cells might be engineered directly within the patient using gene delivery tools, perhaps specialized nanoparticles or modified viruses that specifically target T-cells and deliver the CAR gene, effectively turning the patient's own body into the bioreactor.
Wow, that would be a game changer.
It really would. Researchers are also developing bioinstructive materials. These are smart materials like specialized gels or scaffolds designed to guide and enhance CAR T-cell generation outside the body, but in more efficient, perhaps automated ways. And a truly exciting area is the exploration of rapid CAR T-cell generation techniques. This aims to shorten or even entirely eliminate the initial activation and expansion steps of the T-cells outside the body, which currently take weeks. If successful, this could dramatically cut down the manufacturing time and logistical challenges, potentially making the therapy faster to deliver and more affordable for patients in critical need. The goal is really to move from weeks to days or maybe even hours for production.
Incredible potential there for broadening access. So, putting this all in context, CAR T-cell therapy feels like such a modern breakthrough, almost futuristic, but it clearly builds on centuries of scientific inquiry, right? This idea of how our immune system interacts with cancer. Can you take us way back to the origins of this incredible idea of fighting cancer with immunity? It must have been a very different landscape back then.
Oh, absolutely. This concept is remarkably even anciently old. It predates much of modern medicine, certainly modern immunology. Early insights, or perhaps just observations, stem from as far back as ancient Egypt, maybe 3,000 years ago. There are texts noting spontaneous tumor regression sometimes observed after severe infections and high fever.
Really? Ancient Egypt?
Yes. These were, of course, anecdotal observations, not scientific studies, but they planted a tiny seed of an idea that the body's own defenses, perhaps stimulated by infection, might play a role in fighting tumors. Fast forward to the Greek physician Galen, who lived around 2,000 years ago. He speculated on the role of inflammation in cancer. He observed that tumors often seem to arise in regions of previous injury or chronic inflammation. His term oncos for tumors actually still persists in our terminology today, oncology. His hypothesis, though limited by the medical understanding of his era, laid an early foundation for exploring the links between the body's response and cancer development.
Inflammation as a potential trigger or link.
Right. Then moving into the 19th century, scientific understanding began to evolve more rapidly, with more detailed observations. In 1828, the French surgeon Jean-Nicolas Marjolin noted the development of squamous cell carcinoma, specifically in old burn scars, suggesting that chronic irritation could lead to cancer. An English surgeon, Caesar Hawkins, corroborated this observation in 1833, noting similar cases. But it was Rudolf Virchow, a really key figure in pathology in the late 19th century, who made a truly significant contribution, pointing more directly towards the immune system. He actually identified leukocytes, white blood cells, within tumor tissues under the microscope.
Immune cells in the tumor.
Exactly. This led him to propose the groundbreaking theory that tumors could originate from areas of chronic inflammation, further strengthening the link. He essentially saw the potential battlefield within the tumor itself.
And then came a truly pivotal figure, someone who really put the immune in immunotherapy, even if his methods were met with, well, considerable skepticism at the time. Sounds like a story of a lone innovator ahead of his time.
That would indeed be Dr. William B. Coley in the late 19th century, specifically around 1891. He is often rightly regarded as the father of immunotherapy for his pioneering work. Coley was a bone sarcoma surgeon, and he observed a fascinating and hopeful phenomenon. spontaneous tumor regression in some of his cancer patients following erysipelas.
Erysipelas, that's a skin infection, right?
Yes, a streptococcal skin infection that caused high fever. He hypothesized, quite intuitively for the time, that the strong immune response triggered by the infection might somehow be attacking the cancer cells as well.
So harnessing the response to infection.
Precisely. Driven by this observation, Coley began experimenting. He created a mixture of heat-killed streptococcal bacteria and another bacterium, Serratia marcescens. This combination became famously known as Coley's toxins. He injected this mixture directly into patients' tumors, primarily bone and soft tissue sarcomas.
Injecting bacteria into tumors. That
Yeah.
sounds risky.
It was, by today's standards, certainly. But remarkably, he achieved over a 10% cure rate in some of his patients, a truly extraordinary feat for that era, when options were virtually non-existent. His work was revolutionary, but yes, it faced significant skepticism from the established medical community. Partly because the underlying mechanisms weren't understood, and partly because of the risks. The Journal of the American Medical Association even published a statement in 1894 condemning the use of his toxins, calling them an entire failure.
Ouch.
Yeah. And decades later, in 1963, the FDA reclassified Coley's toxins as an investigational drug, basically halting its wider use due to a lack of contemporary safety and efficacy data, despite it having been used for over 70 years.
So it fell out of favor.
It did for a long time. However, Coley's fundamental principle that certain cancers respond positively to an augmented immune response was later profoundly validated by modern science. His intuition really paved the way for modern approaches like oncolytic virus therapies, which use viruses to stimulate an immune response against cancer. So his early work, despite the skepticism, left a powerful legacy.
So, despite those early hurdles and the skepticism Coley faced, the profound idea of immune surveillance, this concept that our bodies are constantly looking for cancer cells, almost like an internal security system, began to truly take shape. It sounds like a natural extension of Coley's intuition, but maybe with a more theoretical framework.
Exactly. In the early 20th century, specifically 1909, Paul Ehrlich, another giant in early immunology, introduced the explicit concept of immune surveillance. He suggested that the immune system continuously monitors the body for abnormal cells, including cancerous ones, and eliminates them before they become a problem.
The immune system as a guardian.
Yes. This hypothesis marked a major conceptual shift, providing a theoretical foundation, though the experimental tools to really verify it were lacking at the time. Later, Lewis Thomas refined this idea with the term immunosurveillance, proposing that the immune system detects newly developing tumors by recognizing tumor-specific neoantigens.
Neoantigens. New antigens on the tumor cells.
Correct. Novel antigens expressed by tumor cells due to their mutations. Thomas suggested the immune system recognizes these, much like how it recognizes and rejects foreign tissue grafts. The immune system's specific ability to actually elicit an anti-tumor response was first clearly demonstrated experimentally by Ludwig Gross in 1943. He successfully immunized mice against a sarcoma, showing that adaptive immunity could indeed be trained to target tumors. These pioneering ideas really forms the theoretical bedrock for much of modern immunotherapy.
The 20th century then brought even more targeted approaches, moving beyond just general immune stimulation to more precise tools, like the development of monoclonal antibodies. How did these change the game?
Indeed, monoclonal antibodies were a huge leap forward. These are antibodies engineered in the lab, characterized by their exquisite specificity. They bind to only one single, carefully chosen target on cells. This offered a way to deliver very precise attacks against cancer cells, expressing a specific marker, marking a significant step towards targeted therapy.
More like a sniper rifle than a shotgun.
Exactly. The FDA approval of rituximab in 1997 was a watershed moment. Rituximab is a monoclonal antibody specifically designed to target the CD20 antigen, which is expressed on many lymphoma cells. Its approval marked one of the first successful integrations of these highly specific antibodies into mainstream precision medicine for cancer. This milestone not only provided a new, effective treatment for lymphoma, but also really opened the floodgates for the intensive exploration and development of numerous other antibody-based treatments for a wide range of cancers. Today, monoclonal antibodies are an absolutely integral and powerful part of our therapeutic arsenal.
Right. And then we arrive at the really recent Nobel Prize-winning breakthroughs that truly unlocked modern immunotherapy as we know it today, especially this concept of immune checkpoints. These sound like the brakes of the immune system.
Yeah.
How did understanding these revolutionize cancer treatment?
This leads us directly to the pivotal discoveries of James P. Allison and Tasuku Honjo, who were jointly awarded the 2018 Nobel Prize in Physiology or Medicine, a huge recognition. Their groundbreaking work was recognized for their discovery of cancer therapy by inhibition of negative immune regulation. Bit of a mouthful, but crucial.
Inhibition of negative immune regulation, so releasing the brakes.
Precisely. Their insights essentially added a new pillar to the existing foundational cancer treatments, surgery, radiotherapy, and chemotherapy. They fundamentally changed how we approach cancer by realizing we could unleash the immune system's own power rather than just trying to boost it externally or poison the cancer cells.
Okay, tell us about these brakes.
All right, first let's look at CTLA-4, or cytotoxic T lymphocyte antigen 4. This molecule was cloned back in 1987. Initially, its precise function wasn't clear, but later, in 1994, it was decisively determined to be a negative regulator of T-cell activation. Think of it as a crucial brake pedal for the T-cells, preventing them from overreacting and causing autoimmune damage to healthy tissues. A vital safety mechanism.
Right, you need brakes.
You do. The significance of this brake was profoundly confirmed when inactivation of the CTLA-4 gene in mice led to severe, fatal autoimmune disease. This clearly showed its critical role in suppressing immune responses. Now, James Allison, with his unique foresight, took a radical approach. Instead of trying to enhance T-cell activation directly, he sought to block the CTLA-4 brake. He believed that by doing so, he could truly unleash the immune system's inherent power against cancer cells, which often exploit these brakes to hide.
A counterintuitive idea at the time.
Very much so. His pioneering experiments, conducted at the end of 1994 and published in 1996, used antibodies specifically designed against CTLA-4 in tumor-bearing mice. The results were truly spectacular. It showed curative potential. Not only did blocking CTLA-4 strengthen the T-cell anti-tumor response, but even pre-established tumors were sensitive. Rejection led to durable immunity, meaning the mice were protected from getting the same cancer again. And various tumor types responded: colon, prostate, mammary, melanoma. This was the birth of what we now call immune checkpoint inhibitors, or ICIs, a whole new class of drugs. The major clinical breakthrough followed with a phase 3 trial for metastatic melanoma, leading to the FDA and EMA approval for the first anti-CTLA-4 antibody, known as ipilimumab, in 2011. This really marked the shift from the lab bench to the patient's bedside.
Okay, so CTLA-4 was the first major checkpoint target. What about PD-1?
Right. In parallel, PD-1, or programmed cell death protein 1, was identified by Tasuku Honjo's group in Japan in 1992. Initially, its name came from the thought it was involved in apoptosis or programmed cell death. However, it was later discovered that PD-1 also acts as another crucial brake on T cells, though it works through a slightly different mechanism than CTLA-4. You can think of PD-1 as interacting with a don't attack me signal, called PD-L1, that cancer cells often display on their surface to evade the immune system.
Ah, different kind of shield for the cancer. It's exactly. Studies in 2002 showed that tumor cells expressing PD-L1 could cunningly evade immune attack. And critically, this evasion could be reversed by blocking either PD-1 on the T cell or PD-L1 on the tumor cell with specific antibodies. Honjo's subsequent studies in mice, around 2005, then showed that PD-1 blockade could be just as or even more efficient than CTLA-4 therapy in treating tumors. And importantly, it seemed to come with less severe autoimmune side effects.
More effective and potentially safer? That's significant.
Very significant. These remarkable discoveries quickly translated into clinical trials. A pivotal phase I study for the PD-1 antibody nivolumab started in 2006 and showed it was well tolerated. 2 years later, significant efficacy was reported in a larger phase I study across different advanced cancers, including lung cancer, melanoma, and renal cancer. Objective responses were seen in 20–25% of patients, many of which were durable, lasting a long time, with some even leading to complete tumor regression.
Although it's important to note, severe immune-related adverse events, mainly pneumonitis or lung inflammation, did occur in about 14% of patients. Concurrently, another PD-1 antibody, pembrolizumab, also showed very positive results in melanoma patients, with clinical responses in about 38% of patients. Crucially, some of these responses were seen in patients who had not responded to or had stopped responding to the anti-CTLA-4 treatment, suggesting these drugs work differently and could benefit different patients.
So, multiple options emerging.
Yes. This led to rapid FDA approval in 2014 for both pembrolizumab and nivolumab for unresectable or metastatic melanoma, with subsequent expansions to many other cancers, including non-small cell lung cancer, becoming major players in oncology.
And what about combining these therapies, blocking both brakes at once?
That's become a key strategy. Critically, combining therapies that block both the CTLA-4 and PD-1 pathways appears to induce an even stronger anti-tumor effect in some cancers. This has been particularly observed in melanoma and renal cell carcinoma, for example. The thinking is that these two checkpoints operate through different yet complementary mechanisms of action. So blocking both hits, the immune system's brakes at multiple points, leading to a more robust anti-tumor response.
A synergistic effect?
Potentially, yes. This multi-pronged approach often yields superior outcomes in terms of response rates and survival for some patients. Although, understandably, it can also increase the frequency and severity of those immune-related side effects, which is a complexity doctors are constantly learning to manage better.
Okay, so these therapies, CAR T cells, checkpoint inhibitors, are incredibly powerful. But every powerful treatment comes with its own set of considerations, potential downsides. CAR T cell therapy in particular sounds like an incredibly intense process. What are the main side effects patients need to be aware of, and how are those managed?
You're absolutely right. It is an intense process, and managing the side effects is a critical part of CAR T-cell therapy. While incredibly powerful, it does come with significant potential toxicities that patients and their care teams need to monitor very closely. The two most common and serious side effects are cytokine release syndrome, CRS, and neurological issues, sometimes called ICANS, or immune effector cell-associated neurotoxicity syndrome.
Cytokine release syndrome. You mentioned cytokines are inflammatory signals.
Yes, exactly. CRS is essentially a profound inflammatory cascade. When the newly infused and activated CAR T cells begin multiplying and attacking cancer cells, they effectively go to war inside the body. In doing so, they release a flood of these inflammatory proteins, the cytokines. This can lead to a systemic inflammatory response, almost like a severe body-wide flu, but potentially much more dangerous.
What are the symptoms?
Symptoms of CRS can range from relatively mild, like fever, chills, fatigue, to very severe. This can include dangerously low blood pressure, difficulty breathing or shortness of breath due to fluid in the lungs, and in rare, severe cases, it can lead to serious heart or lung failure requiring intensive care. It's paradoxically a sign that the engineered cells are actively working and killing cancer, but it needs incredibly careful, often rapid medical management.
How is it managed?
Often with supportive care like fluids and oxygen, but also specific medications that block key cytokines like IL-6 inhibitors such as tocilizumab, which can often rapidly reverse severe CRS.
Okay, and the neurological issues? ICANS.
Right. Beyond CRS, patients can also experience a range of neurological issues, or ICANS. The exact cause isn't fully understood, but seems related to the inflammation crossing into the brain. These can manifest in various ways, sometimes quite frighteningly for patients and families. Things like difficulty speaking or forming words, known as aphasia, or problems with balance and coordination. Patients might also experience confusion, disorientation, difficulty writing, dysgraphia, severe headaches, seizures, tremors, or involuntary muscle twitching.
That sounds serious.
It can be. These neurological toxicities, like CRS, are typically temporary and reversible, but they require prompt medical attention and supportive care. Often, corticosteroids are used to reduce inflammation in the brain. Close monitoring is essential.
Are there other significant side effects besides CRS and ICANS?
Yes, another significant potential side effect is what we call on-target off-tumor recognition.
On target off tumor, meaning it hits the right target but on the wrong cells.
Exactly. This occurs when the CAR T cell, designed to recognize a specific antigen on cancer cells, also finds that same antigen expressed, perhaps at lower levels, on healthy non-pathogenic tissue in the body. A classic and well-understood example involves CAR T cells targeting the CD19 antigen. CD19 is found on many B-cell blood cancers, which is great. But it's also found on normal, healthy B cells, which are crucial for producing antibodies and fighting infection.
Ah, so the CAR T cells wipe out the healthy B cells too.
Precisely. This leads to a condition called B-cell aplasia, meaning a significant depletion or even complete absence of healthy B cells in the body. This on-target off-tumor effect, combined with a patient's prior immunosuppression from their disease and the lymphodepleting chemotherapy they received before the CAR-RT infusion, places patients at a significantly increased risk of serious infections.
Because their antibody production is compromised.
Correct. Their immune system is weakened, making them more vulnerable to common bacteria and viruses. They often need immunoglobulin replacement therapy long-term. Furthermore, they can experience low counts of various other blood cells like neutrophils or platelets for extended periods, a condition called prolonged cytopenias, requiring careful monitoring and sometimes transfusions or growth factor support.
It's a lot to manage.
It is. And in addition to these major concerns, other potential side effects patients might experience include an allergic reaction during the infusion itself. a pervasive feeling of fatigue that can last for weeks or months, bruising or bleeding more easily than usual due to low platelets, and electrolyte imbalances like low potassium or sodium, which need correction. Given the potential severity and complexity of all these risks, patients typically need that hospital stay of 7 to 10 days, sometimes longer, post-infusion for intensive monitoring by specialized teams trained in managing these unique side effects. And as we mentioned, even after discharge, they need to stay close to the center, have a caregiver, and avoid driving for a while. The care required is just as intense as the therapy itself.
Right. And briefly, what about the side effects of the immune checkpoint inhibitors we discussed earlier, like ipilimumab or nivolumab? Are those generally different?
Yes, while also immune-related, the side effects from immune checkpoint inhibitors, the ICIs, are generally distinct from those seen with CAR T-cell therapy, largely because of their different mechanisms. Remember, ICIs broadly unleash the entire immune system by removing its natural brakes, whereas CAR T-cells are specifically engineered soldiers targeting one thing.
So ICIs can cause broader autoimmune reactions.
Exactly. Because the immune system is no longer fully controlled by these brakes, ICIs can cause a wide range of severe immune-related adverse events. Essentially, autoimmune-like reactions, where the now unleashed immune cells inadvertently attack healthy tissues anywhere in the body. For instance, with CTLA-4 inhibition, like ipilimumab, severe inflammation of the colon, colitis, can occur. Or endocrine dysfunction affecting hormone production issues with the pituitary, adrenal glands, or thyroid are not uncommon. Thyroiditis can happen with both CTLA-4 and PD-1/PD-L1 blockers. Pneumonitis, that inflammation of the lung tissue, is mainly associated with anti-PD-1/PD-L1 therapies. In some instances, these adverse reactions can be fatal if not recognized and managed promptly and aggressively, often with high-dose steroids to suppress the immune system again.
So different profiles, but still serious potential side effects requiring vigilance.
Absolutely. However, many are treatable. For example, hydrocortisone supplementation can effectively treat adrenal insufficiency caused by these therapies. The key, as with SIRA T-cell therapy, is vigilant monitoring by experienced teams and rapid intervention to manage these unique immune-related toxicities.
This all sounds like incredibly advanced, complex medicine, requiring significant resources, specialized centers, expert teams. I can imagine the cost is equally significant, potentially putting it out of reach for some. How does the healthcare system grapple with the economics of such a revolutionary yet expensive treatment?
You're absolutely correct in your assumption. The cost of SIRO T-cell therapies has indeed been a significant point of discussion, even criticism, globally. It's largely due to the immense complexity involved at every single stage. When these treatments were first introduced, the list prices were staggering. Initial costs for therapies like Kymriah were around $375,000 for a single treatment for L. For Yescardi, for lymphoma, it was about $475,000.
Just for the therapy itself.
Just for the bag of cells, yes. Those figures typically don't include the extensive associated medical care, the leukapheresis, the bridging therapy, the lymphodepleting chemo, the lengthy hospital stays, the intensive monitoring, managing side effects like CRS or ICANS, follow-up care. All of that can easily add hundreds of thousands more to the total cost of care per patient.
What drives those high costs?
Several factors. Firstly, there's that intricate, highly personalized cellular manufacturing process itself. It takes place in highly specialized good manufacturing practice, or GMP facilities. These are custom, ultra-sterile environments requiring immense technological investment and highly skilled personnel to safely and precisely re-engineer each patient's cells, ensuring quality and preventing contamination. It's bespoke medicine at its most complex.
Right, it's not mass-produced.
Exactly. Secondly, there's the high level of intensive hospital care necessary after the CAR T-cells are administered. As we discussed, patients face those serious risks like CRS and neurotoxicity. This requires close, specialized monitoring by experienced multidisciplinary teams, often involving stays in intensive care units and frequently prolonged inpatient stays. All of this contributes substantially to the overall cost.
So how is it covered? Is it accessible?
It's a challenge. In the U.S., Medicare does cover AR T-cell therapies for approved indications. Many, though thankfully not all, private insurers also provide coverage. However, navigating approvals and potential out-of-pocket costs can still be a significant hurdle for patients. Recognizing this, manufacturers of AR T-cells have proactively developed some alternative, innovative payment programs to try and address the high upfront cost.
Like what?
For example, some early programs involved outcomes-based agreements. They might require payment from the insurer or hospital only if the CAR-T therapy induces a specific response, like a complete remission, by a certain time point after treatment.
Pay for performance, essentially.
Essentially, yes. It aligns the cost more directly with successful patient outcomes and shifts some of the financial risk from the payer to the manufacturer. It's an evolving area, trying to balance the high investment needed for these transformative treatments with the need for patient access and healthcare system sustainability.
Looking ahead then, where is this all going? This field is clearly not standing still. The pace of innovation seems incredibly rapid. What are the major frontiers researchers are currently pushing?
Not standing still at all. The future of cancer immunotherapy, particularly cell therapy, is brimming with immense promise and constant evolution. Researchers worldwide are deeply invested in expanding CAR T-cell therapy beyond its current, somewhat limited focus on certain blood cancers. They're aiming to tackle much broader challenges, especially solid tumors.
Right, the solid tumor challenge. You've mentioned that's a big hurdle.
It is. A major ambitious goal is to make CAR T-cell therapy truly effective against solid tumors. These, remember, account for about 90% of all adult cancers and have historically been notoriously difficult to treat with CAR T-cells. The challenges are formidable. Solid tumors often exhibit that antigenic heterogeneity we talked about. The cancer cells within a single tumor can have a diverse range of antigens, making it hard for a single SAR design targeting one thing to eliminate them all. Escape is easier.
The cancer cells can adapt or hide.
Exactly. Plus, solid tumors exist within a highly immunosuppressive tumor microenvironment, the TME. This local environment actively works to suppress immune cell activity and protect the cancer cells. It's like a hostile fortress for the T-cells. Furthermore, issues like T-cell exhaustion are significant hurdles. The CAR T-cells can basically get worn out and lose their potency over time when fighting constantly in this hostile tumor environment.
So how are researchers trying to overcome these challenges?
There's exciting progress on multiple fronts. For instance, tackling heterogeneity. Researchers are now using CAR T-cell technology to simultaneously target two different antigens on glioblastoma tumor cells. Glioblastoma is one of the deadliest and most aggressive brain cancers.
This dual targeting.
Yes, this multi-antigen targeting offers new hope by making it much harder for cancer cells to escape destruction simply by losing one antigen. Another example, in breast cancer, specifically triple-negative breast cancer, which is particularly aggressive and difficult to treat, an innovative approach uses a gel infused with CAR T-cells.
A gel, how does that work?
This special hydrogel, loaded with CAR T-cells, can be delicately applied directly to the surgical site right after a tumor has been removed. The idea is to target and eliminate any microscopic residual cancer cells that might have been left behind, preventing recurrence. Preclinical studies in mice have shown outstanding effectiveness in eliminating cancer without compromising normal tissue healing. And clinical trials are now underway exploring direct infusion of CAR T-cells into tumors, marking a significant step toward making CAR-T viable for solid tumors.
That's fascinating, local delivery right where it's needed.
Exactly. And what's also truly fascinating is that the application of CAR-T cell therapy or similar engineered cell therapies is being explored for non-cancerous conditions too.
Really? Beyond cancer?
Yes. This truly highlights the versatility and foundational nature of this technology, reprogramming immune cells to target specific problems. It's being investigated for autoimmune diseases like systemic lupus erythematosus or lupus. The idea is to design CAR T cells to target and eliminate the specific B cells that are mistakenly producing autoantibodies and causing the disease, potentially offering a way to reset the immune system. Early results have been very promising.
Targeting the cause of autoimmunity.
Potentially. And in a really remarkable step, researchers at Penn Medicine have developed an innovative technique combining CAR T-cell principles with mRNA technology to address cardiac damage, specifically heart failure caused by fibrosis or scarring.
CAR-RT for heart failure. How?
They're not actually using permanent CAR T-cells here, but temporarily reprogramming T-cells using mRNA similar to the COVID vaccines to make them target heart fibroblast cells. These are the cells responsible for producing scar tissue after heart injury, which leads to heart failure.
So temporary targeting of scar-forming cells.
Exactly. Early trials in mouse models of heart failure show that these transiently reprogrammed T cells effectively reduce the number of cardiac fibroblasts, leading to a significant reversal of fibrosis and an improvement in heart function. This innovative approach holds immense promise for developing entirely new therapies to potentially reverse heart damage, expanding the scope of engineered cell technology far beyond oncology.
That's incredible potential. Now, beyond the traditional CAR T cells we've discussed, are there other next generation cell therapies on the horizon, maybe using different types of immune cells?
Absolutely. The versatility of this cellular engineering approach is truly inspiring, and the pipeline of new cell therapies is incredibly robust. Researchers are exploring other immune cell types. Promising candidates include CAR-M, CAR macrophages, and CAR-NK, CAR natural killer cells.
Macrophages and NK cells, they have different roles in the immune system, right?
They do. Macrophages are phagocytic cells, meaning they can engulf and digest targets, and they are very good at infiltrating dense tissues, which could be an advantage in solid tumors. NK cells are innate immune cells that can kill targets without prior sensitization. Engineering these alternative cell types with SCARs is showing positive results in preclinical and early clinical studies. They might overcome some limitations of T cells, offering different ways to attack cancer or navigate the tumor microenvironment. And in the broader field of adoptive T cell therapy, ATT, which involves isolating, expanding, and then reinfusing a patient's own naturally occurring cancer-fighting T cells, typically those found within the tumor itself, tumor-infiltrated lymphocytes or TILs, we've seen major advancements.
GIL therapy, that's been around for a while conceptually, hasn't it?
It has, pioneered by Dr. Steven Rosenberg at the NIH, but making it a standardized approved therapy has been a long road. But just recently, in February 2024, Lifileucel received U.S. FDA approval. This is a monumental step. It marks the first TIL-based cellular therapy approved for a solid cancer, specifically advanced melanoma. This really expands the reach of personalized cellular immunotherapy beyond blood cancers into the solid tumor realm. It's a huge validation for the ACT field.
That is a major milestone. It sounds like understanding the tumor's environment itself, the TME, is also absolutely key to making all these therapies work better in the future. It's not just about the engineered cell.
It is absolutely critical. The tumor microenvironment, TME, is now recognized as a major player. It's not just the cancer cells, it's the whole ecosystem surrounding stromal cells, blood vessels, signaling molecules, and crucially, other immune cells. This TME plays a crucial and often detrimental role in cancer progression, and notably in determining how well a tumor responds or resists therapy.
So future efforts need to tackle the TME too.
Definitely. Future strategies increasingly involve precisely modulating this TME to enhance immunotherapy effectiveness. For example, researchers are working on ways to counteract or deplete the immunosuppressive cells that often accumulate within the TME. These include regulatory T cells, Tregs, myeloid-derived suppressor cells, MDSCs, and certain types of tumor-associated macrophages, TAMs. These cells actively suppress the anti-tumor immune response, essentially creating a shield around the cancer that therapies like CAR T cells have to penetrate.
Finding ways to disable the shield.
Exactly. Tumors also employ other clever tactics, like releasing exosomes, remember those tiny vesicles?
Right, the little packages they send out.
Yes, they can release exosomes packed with a cargo of immunosuppressive molecules,
Wow.
things like TGF-beta, IL-10, even PD-L1 itself, and specific microRNAs. These exosomes can travel and inhibit immune cell function nearby or even systemically promote immune evasion and cause dysfunction in T cells or NK cells. They're like tiny propaganda messengers suppressing the immune response.
Wow.
Understanding and counteracting these sophisticated mechanisms, as well as addressing metabolic reprogramming within the TME, where cancer cells alter their metabolism to outcompete immune cells for nutrients like glucose, are key areas of intense research focus.
It's incredibly complex. What about factors outside the tumor, like the microbiome?
That's another fascinating and relatively recent area. The microbiome, the vast community of microorganisms living in and on our body, particularly in the gut, has been discovered to significantly influence cancer treatment outcomes, including how well a patient responds to immunotherapy, especially checkpoint inhibitors.
The gut bacteria affecting cancer treatment.
It seems so. The composition of the gut microbiome appears to influence the systemic immune tone. So manipulating the microbiome, perhaps through approaches like specialized probiotics, prebiotics, or even fecal microbiota transplantation, FMT, is being explored as a novel strategy to potentially improve patient responses to immunotherapy. It's a surprising connection, but potentially powerful. And all these efforts really tie into the broader ultimate goal of personalized medicine. This involves identifying specific biomarkers characteristics of the patient or their tumor, like the tumor mutational burden, how many mutations the cancer has, the expression levels of PD-L1, or specific genetic features like microsatellite instability, MSI. These biomarkers can help predict how a patient's cancer is likely to respond to a particular immunotherapy. This allows oncologists to select the most appropriate, tailored treatments for each individual, moving away from a one-size-fits-all approach and ultimately leading to much better, more predictable outcomes and sparing patients from ineffective therapies and their side effects.
It's truly a dynamic field. You mentioned over 1,000 clinical trials for TRT cell therapies alone are currently underway. That's incredible activity.
It is. Each one pushing the boundaries, testing new designs, new targets, new combinations, tackling new diseases. It's constantly revealing new layers of complexity, but also immense opportunity. Hashtag outro.
Well, our deep dive into CAR T-cell therapy has taken us on quite an incredible journey today. From the microscopic world of T-cells and antigens, through the pioneering spirit of scientists like William Coley, who dared to think differently centuries ago, to the Nobel laureates James Allison and Tasuku Honjo, whose discoveries really reshaped our understanding and treatment of cancer in the modern era. We've explored the intricate, multi-step, highly personalized process of creating these living drugs. We've understood their current profound impact, particularly on certain blood cancers, and we've glimpsed the exciting future where they might tackle the formidable challenge of solid tumors and even be repurposed to treat non-cancerous conditions like autoimmune diseases and potentially even cardiac damage.
It's very clear that immunotherapy, with CAR T-cell therapy and related cell therapies at its forefront, represents a monumental shift in how we combat disease. It really is a true new pillar of cancer treatment. It's offering real hope and the potential for longer, healthier lives to many patients who previously had very limited or sometimes no effective options left. The depth and breadth of the ongoing research just underscore the profound belief in this approach and its future potential.
This deep dive has truly showcased the fascinating intersection of immunology and oncology, revealing how our own bodies hold these incredibly powerful keys to fighting even the most complex diseases. The ingenuity in harnessing these natural defenses and then refining them with modern science like genetic engineering is just nothing short of revolutionary.
It really is. And this raises an important, maybe even profound question for us all to consider as we look forward. As we continue to get better at harnessing the body's own incredible power to heal and fight disease, what other conditions, beyond cancer, beyond autoimmune disorders, beyond cardiac issues, might someday be transformed by these living, personalized cellular therapies? The possibilities seem to be just beginning to unfold, inviting us to imagine a future where our own cells truly become the ultimate medicine.
A fascinating thought to end on. Join us next time for another deep dive into the fascinating world of knowledge.